Dear students, please note down. These notes are available in the description of video. Please download your notes. In today's lecture, we will discuss biogenesis or origin of Golgi body, Golgi complex, dictyosome. As well as in today's lecture, we will clear different functions of Golgi complex. In our last lecture, we have studied ultrastructure, electron microscopic structure of Golgi complex, Golgi body. And that structure shows this Golgi complex consists of 4 components: cisterna, tubules, secretory vesicles, and vacuoles. In today, we will clear biogenesis, means origin of Golgi complex. There are different views suggested by different scientists regarding the origin of biogenesis of Golgi complex. Some scientists believe that this Golgi body, it is derived from the vesicles which develop from plasma lemma, cell membrane, while some scientists believe that this entire Golgi complex is derived from the vesicles which are produced by the outer nuclear envelope, ectokaryotheca, and these small vesicles may get fused to produce annulated lamellae. Either these vesicles or these annulated lamellae give rise the Golgi complex. But the most accepted view regarding biogenesis, regarding origin of Golgi complex, that was suggested by Novikov et al. In 1962. And this view regarding its biogenesis suggests that rough endoplasmic reticulum, which is responsible for the formation of, means the Golgi complex is evolved from endoplasmic reticulum, ER. Rough endoplasmic reticulum, that RER gives rise the smooth endoplasmic reticulum due to loss of ribosomes. Rough endoplasmic reticulum having ribosomes, and due to the loss of ribosomes, the rough endoplasmic reticulum changes into smooth endoplasmic reticulum. And this smooth endoplasmic reticulum, it gives rise small bud-like structures, blab-like structure, either due to budding or blabbing. Small vesicles are formed, and these vesicles are named transition vesicles. And these transition vesicles are responsible for the formation of these cisterna of Golgi body towards the cis end, forming end of Golgi complex. This is the interrelationship between different cell membranes that was suggested by Novikov. According to this view, this outer coat of nucleus ectokaryotheca, and this ectokaryotheca, it gives small external buds. These are blabs or buds. Due to budding or blabbing of this ectokaryotheca, small vesicles are formed. And these vesicles fuse to produce a lamellae, and these are named annulated lamellae. Note down, this outer coat of nucleus, that is ectokaryotheca, and this ectokaryotheca, it gives rise small outbuds. These external buds, or these are bulb-like structures, due to budding or blabbing, these small vesicles are formed. And these vesicles fuse to produce annulated lamellae. From these annulated lamellae, endoplasmic reticulum, means first of all rough endoplasmic reticulum develops. And from these rough endoplasmic reticulum, smooth ER, smooth endoplasmic reticulum appears. And the budding or blabbing of smooth endoplasmic reticulum, small vesicles are formed. These are transition vesicles, these are forming vesicles, and these vesicles fuse to produce cisterna of Golgi body at cis phase or forming phase of Golgi complex. This is interrelationship between different cell membranes, as well as this plasma lemma also gives small vesicles, and these vesicles, some scientists believe that these plasma lemma vesicles are responsible for the formation of Golgi complex. These are different functions of Golgi body. Very important topic. First of all, ultrastructure of Golgi complex. And that ultrastructure shows its 4 components. These fine sac-like structures, these are cisterna. From the terminal parts of these cisterna towards periphery, fine tubular network develops. The second part of Golgi complex, tubules. And from the ends of tubules, secretory vesicles are formed. And these, the swollen parts of these cisterna give rise large sized vacuoles which act as primary lysosomes. These are different functions of Golgi body. Now NEET students, competitive exam students. First, very important function is cell secretion. And that cell secretion mainly consists of glycoproteins. Note down the glycoproteins. And these glycoproteins are complex molecules made up of carbohydrates and proteins. Formation of glycoproteins. The rough endoplasmic reticulum. This rough endoplasmic reticulum which possesses ribosomes, and these ribosomes are involved in the synthesis of proteins. This process of formation of proteins by ribosomes, that is called translation. Rough endoplasmic reticulum on which ribosomes are present, and these ribosomes are protein factories which involve in the synthesis of protein. That process is translation. And the proteins which are formed by the ribosomes enter here in the lumen of rough endoplasmic reticulum cisterna. Here in the form of transition vesicles. Note down, the proteins which are synthesized by ribosomes enter in the lumen of rough endoplasmic reticulum, and from these rough endoplasmic reticulum, these protein molecules, these enzymes, proteins which are further passed to small vesicles, these are transition vesicles. Through these transition vesicles, the proteins enter here in the cisterna, in the lumen of cisterna of Golgi body. Here in Golgi body, the proteins which are derived from rough endoplasmic reticulum here enter in the lumen of cisterna of Golgi body. Here, these proteins interact with carbohydrates like mucopolysaccharides to produce glycoproteins. Note down the glycoproteins, these are complex molecules. These are made up of carbohydrates plus proteins. The proteins which interact with heteropolysaccharides, mucopolysaccharides, and the process is named glycosylation. The process of formation of glycoproteins means carbohydrate plus proteins, the proteins which are derived from the rough endoplasmic reticulum, and the carbohydrates which are present here in the Golgi body to produce glycoprotein. The process is glycosylation, and this reaction is catalyzed by glycosyl transferase enzyme. Very important property, that is cell secretion. The Golgi body, the main function of Golgi body, that is cell secretions. The hormones which are produced by endocrine cells, the antibodies, the immunoglobulins produced by plasma cells, the neurotransmitters which are produced in nerve cells, neurons, the collagens which are produced in connective tissue, all these secretions are produced by Golgi complex. It means the main function of Golgi complex is cellular secretions. The mucus, the hormones, the collagen, the neurotransmitters, the hormones, the proteins, the enzymes, the lipids. These macromolecules are transported inside the cell through the Golgi body. Note down the proteins which are synthesized here by rough endoplasmic reticulum. These proteins enter here in Golgi body through these transition vesicles, and here in Golgi body the packaging of macromolecules are packed here, transformation here as well as condensation. Note down this Golgi body also involves in condensation, in packing, transformation and secretion of macromolecules. It means this Golgi body acts as a mediator between this endoplasmic reticulum and cell membrane, as well as it acts as a traffic police of the cell. Note down very important function, very important property of Golgi complex. It involves in cell secretions. Dear students, cell secretions, whether these are hormones, neurosecretory cells, neurons which produce neurotransmitters, the collagen produced by the connective tissue, all secretions, the mucus by goblet cells, and these secretions are produced by Golgi complex. And this Golgi complex acts as a mediator between this endoplasmic reticulum and cell membrane, as in Golgi body, which acts as a traffic police of the cell, because it involves in packaging of molecules, it involves in the condensation of, it involves in the transformation of and secretion of molecules in the form of secretory vesicles. Here in these secretory vesicles, the macromolecules are packed in the form of granules named secretory granules, and these secretory granules fuse with the cell membrane, plasma membrane, to pass these macromolecules through exocytosis or reverse pinocytosis. Very important function of Golgi complex. It means Golgi body acts as a condensation membrane. The biomolecules, the macromolecules which are produced inside the cells are transported through this Golgi body. This Golgi body acts as a mediator between this ER and cell membrane, as well as this Golgi body which involves in cell secretion, in packaging of molecules, in condensation of molecules by losing water, as well as transformation of macromolecules and their secretion. So it acts as a traffic police of the cell. Another important property. This Golgi complex involves in the synthesis of hormones, in the synthesis of enzyme, in the synthesis of lipid, metabolism of lipid, and dear students, in the secretion of antibodies, in the secretion of neurohormones, in the secretion of neurotransmitters, in the secretion of hormones, etc. Formation of acrosome. This is the diagram of sperm. And its anterior-most part, this anterior-most part of sperm, that is acrosome. This acrosome, it is made up of Golgi body. And this acrosome involves in the secretion of some enzymes like hyaluronidase enzymes. And these acrosomal enzymes acid, acrosomal enzyme, hyaluronidase enzyme, which involve in the dissolution, in the solubilization of egg coats like zona pellucida, corona radiata, vital and membrane of egg. It means at the time of fertilization, this sperm enters in the egg by secreting some acrosomal enzyme like hyaluronidase, and these enzymes, this acrosome which is derived from Golgi body. It means in the formation of acrosome, Golgi body plays an important role. This is another important function of Golgi body. At the time of plant cell division, at the time of cytokinesis, a cell plate is formed between the two daughter cells, the newly formed daughter cells, the cell plate, the middle lamella, the cell wall, and that is produced by both Golgi body and endoplasmic reticulum. The ER as well as Golgi body produce small vesicles, and these vesicles are named phragmoplast. Each and every point very important. These phragmoplast get fused to produce a cell plate, and this cell plate grows outwardly centrifugally to produce first wall, middle lamella, as well as the Golgi body secretes pectic substance for the formation of cell wall. It means at the time of cytokinesis, at the time of cell division in plant cells, the cell wall, the middle lamella is produced by the Golgi body as well as endoplasmic reticulum. Formation of primary lysosomes. The Golgi body also responsible for the formation of lysosomes, primary lysosomes, which contain acid hydrolase enzyme for intracellular digestion. Another important function, vitellogenesis. Note down, vitellogenesis means inside the egg, in the oocytes, egg forming cells, the synthesis of yolk. Note down, synthesis of yolk, the food material, by the Golgi body. Activation of mitochondria. Golgi body is responsible for the activation of mitochondria at the time of ATP synthesis. It means during ATP generation, the mitochondria becomes active, and that stimulus is provided by Golgi body. Formation of pigments in our pigment cells, retina cells. The pigment formation is carried by Golgi body. Formation of root hair. Formation of hypnotoxin. Another important function, formation of hypnotoxin. That is a poison toxic substance which is present in nidoblast, nematoblast, in case of coelentrates, in case of cnidarians, in case of hydra, obelia. For the defense cells, the nematoblast contain toxins, and that toxin is hypnotoxin. Each and every point very important. The Golgi body produces small vesicles between the two newly formed cells, daughter cells. These vesicles are phragmoplasts at the time of cell division, at the time of cell plate formation, during cell division, as well as the Golgi body involves in cell wall formation as it produces pectic substance of the cell wall. In case of sperm, this anterior part which is acrosome, and that acrosome contains hyaluronidase enzyme. The enzymes, the lytic enzymes which involve in the solubilization of egg membrane, egg coats, corona radiata, zona pellucida. It means at the time of fertilization, sperm enters inside the egg due to the acrosomal secretion, and this acrosome is produced by Golgi body. This is Golgi body involves in condensation of biomolecules. Golgi body acts as a traffic police of the cell because the macromolecules, proteins, enzymes, lipid molecules, the macromolecules which are the packing of molecules, transformation of macromolecules, as well as their secretion under control of this Golgi body. And from endoplasmic reticulum to cell membrane, these macromolecules are transported via Golgi body. So it is traffic police of the cell. And this Golgi body acts as an intermediator between this ER and cell membrane. This is interrelationship between different cell membranes. According to Novikov, this outer coat of cell, this outer coat of nucleus, that is ectokaryotheca, it gives small external buds. These are buds, blebs, and these small bulb-like structures give rise vesicles, and these vesicles, which are derived from outer membrane of nucleus, fuse to produce annulated lamellae. These annulated lamellae give rise rough endoplasmic reticulum, and from the rough endoplasmic reticulum, smooth ER are formed, and smooth ER give rise some buds to produce transition vesicles, and these vesicles are responsible for the formation of cisterna of Golgi body. This is interrelationship between different cell membrane as well as biogenesis, origin of Golgi body, that was suggested by Novikov et al. 1962. This is all about Golgi body, ultrastructure, functions, biogenesis. And dear students, in tomorrow's lecture we will discuss ribosomes. Thank you.